Welcome, everyone. We'll get started momentarily. Welcome, everyone, and Jai Jinendra to all of you, and thank you for joining us today for the second inaugural Sri Parshvanath Presidential Chair in Jain Studies Lecture in Ethical Innovations. My name is Brian Donaldson, and I hold the chair in Jain Studies here at UCI in Religious Studies and Philosophy. And I want to welcome all of you here today across many disciplines and locations. I want to send a special welcome to my students in Animal Ethics and Humanities Core, to faculty and students in the School of Medicine, the Schools of Engineering, Biological Sciences, Nursing, and the Humanities, among others, as well as colleagues in the University Laboratory Animal Resources at UCI. I also want to thank James Kyungjin Lee, who directs the Center for Medical Humanities, for hosting this series, and also a special thanks to Joanne Jamora and Sam Carter, also from the center, who have facilitated all the moving parts for an event like this. Lastly, I want to warmly welcome any members of the Jain community here in Southern California, or around the country, or around the world. Welcome. The regional Jain community, in partnership with the Anikant Community Center in La Mirada, have mobilized over the last week to procure and send nearly 6,000 oxygen concentrators to India. If you are interested in supporting their local and global work, I've put the websites here for the Anikant Community Center, as well as for the Indian Red Cross on the screen here. Finally, this lecture is being recorded, and registered attendees will receive a link for the video in about a week's time. And I'd just like to give a few words of introduction for the lecture series. Jainism is a religious tradition and a philosophical system rooted in India, whose texts and practices are perhaps more robustly focused on the ethics of life and death than any other tradition in the world. In fact, the music that you heard playing as you joined today is called the Namakar Mantra. This is a Jain homage to 5 groups of inspiring persons who restrained their mind, their body, and their speech so as not to harm other living beings. Jainism posits a core life force in every living being, whether that be a human, an animal, an insect, or even a microbe. Consequently, the Jain tradition advocates nonviolence, or ahimsa. You can see both the word and the symbol here on the screen, in order to minimize harm to all life forms. This sentiment is expressed in the oldest surviving Jain text, dated back to around the 6th century BCE, which states the following: all breathing, existing, living, sentient beings should not be slain, nor treated with violence, nor abused, nor tormented, nor driven away. This is the pure, unchangeable, eternal law. This powerful exhortation not to harm is expressed in the lives of Jain monks and nuns who seek to avoid violence to all beings in as many ways as humanly possible. Monks and nuns give up their family and their professional life in order to refrain from the accumulation of material goods and wealth, They do not wear shoes. They don't use mechanized transportation. They wear mouth shields to prevent inhaling microorganisms. They eat simple vegetarian food prepared by others. They use a broom to sweep the ground clear of insects when walking, sitting, or lying down, among many other iconic practices. However, the vast majority of Jains in the world are not monks and nuns. They are lay people who strive to live out the commitment to nonviolence in the context of their work, their family, and their communities. Through the tradition's history, lay Jains have gravitated toward occupations that aim to inflict less harm, which came to include medicine. Today, Jains have high representation in medical and allied fields, including the Jains that live here in the US. Today's lecture in ethical innovations emerges from my own research and work with the community regarding Jainism and bioethics, including how Jain medical professionals strive to maximize nonviolence while attempting to ethically manage certain healthcare harms, such as injuring animals through testing, research, and dissection. In teaching bioethics at the university level, many of my students have also expressed their moral struggle with this aspect of their education because they feel deeply committed to reducing harm and to putting forth meaningful research. So rather than silence this difficulty, I believe that we can bring it to the table here at UCI as one of the preeminent research institutions in the world to bring this issue to the light of ethical reflection, and then to join across disciplines to lead visionary grants, projects, fellowships, and partnerships toward developing and implementing alternatives with urgency so that UCI's future of global research can be more effective as we promote a thriving community of humans and other beings whenever possible. As we begin, please note that all of you have a little Q&A icon likely at the bottom of your screen or your image window. You're able to click that and type in a question as it comes to mind throughout the speakers today. Dr. Erin Cariati from the UCI Medical School will moderate our discussion after the presentation and try to tie in as many of your questions as possible. It is now my pleasure to invite Dr. Kunal Parekh to offer a few remarks as a member of the US Jain community. Dr. Parekh is a faculty member at the Center for Nanomedicine at the Wilmer Eye Institute and the Center for Bioengineering Innovation and Design at Johns Hopkins University School of Medicine. There, he leads a team of engineers, scientists, and clinicians working to develop biomedical solutions for significant unmet clinical needs, with a particular focus on ophthalmology. Motivated by his interest in enabling access to healthcare for vulnerable populations, Parekh leads the Global Institute for Vision Equity and previously built and served as executive director for the Social Innovation Lab at Johns Hopkins, where he developed global partnerships and advised dozens of mission-driven entrepreneurs. In 2019, he was included in Forbes 30 Under 30 in healthcare. And I've invited Dr. Parekh to say a few words on the Jain Innovation Network that he recently co-founded and which is sponsoring today's event, as well as his interest in ethical innovation as it relates to Jain nonviolent ethics. Welcome, Kunal.
Thank you for the very kind introduction and such a beautiful introduction to Jainism as well. As you said, my name is Kunal Parekh. I'm a faculty member here at Johns Hopkins, and much of my work focuses on how do we develop novel innovations that can address significant unmet needs globally, and how do we commercialize them in a way that enables access and affordability to the people who need it the most. And much of that is driven as a practicing Jain by my Jain values of serving others and of compassion for others. Recently, we started the Jain Innovation Network with the idea that innovation and entrepreneurship can be really incredible and powerful forces for good. And really that the most successful innovations, the most successful companies are those that really think about all of their stakeholders and want to have a positive impact on all of them, from their employees to their shareholders, to their customers, to where they're sourcing their goods from. The most successful companies are those that are thinking about all of their externalities and trying to improve their impact on all of them. And what we've really thought and learned is that those most successful innovations are essentially those that are in one way or another are leveraging Jain principles of nonviolence, non-stealing, non-exploitation, non-attachment, and maybe foremost of compassion. And so we started the Jain Innovation Network to support the development and support innovators who want to develop innovations, who want to develop companies, keeping those Jain principles in mind. And so I'm personally very excited for the talk today and to be a part of such a wonderful event, and look forward to hearing from the speaker.
Thank you so much, Kunal. It's really great to have you here and to just introduce your important work with the Jain Innovation Network. Now we're going to move on to our main speaker. Kathrin Herrmann is a veterinary expert in animal welfare science, ethics, and law. Since 2017, she has worked at the Center for Alternatives to Animal Testing at Johns Hopkins University School of Public Health, where she directs the Beyond Classical Refinement Program. In this program, she critically appraises current animal use practices in science, with the main focus being teaching the next generation of scientists through international initiatives to improve and extend humane, human-relevant science education. In 2019, Dr. Herrmann initiated and co-edited the open access book, Animal Experimentation, Working Towards a Paradigm Change. And I put the image here on your screen. It's actually an open access book. And so if you just type the title into Brill's website, you'll be able to access a free PDF. The book features 51 authors who critically review current animal use in science using accessible language in order to present innovative non-animal approaches to address urgent scientific questions and offer a roadmap toward a human biology-based science. At the end of 2020, Kathrin took an additional position, and she is now the Animal Protection Commissioner of Berlin, Germany, a role in which she advises the government of Berlin and its citizens in various animal welfare and protection issues. Again, please note that during our talk, you can click on that Q&A box at the bottom of your screen to type in questions as they come to mind, and we'll engage as many as possible at the end. Welcome to UCI, Dr. Herrmann. Over to you.
Brian, thank you so much for the invitation. I feel very honored to be here with you all. And yeah, Jainism has always been very interesting to me. And 6 years ago, when I was in Delhi, I was able to visit a Jain hospital for animals, especially for birds that needed help. And that was quite interesting. So I really enjoyed that. Thank you for having me. I'm going to share my screen now. Can you see that okay?
It is perfectly clear, all looking good.
Okay, great. As you know, I have a little bit spotty internet, so I might go on mute, if not on mute, on off camera, because I think it might be better. I'm a little bit scared because yeah, I wasn't able to get better internet.
Yes, Kathrin is joining us from the Canary Islands right now. So I think, Kathrin, you can start however you'd like. And if you just want to start with your video on, you're welcome to, and I can let you know if there starts to be a lag, or if you'd prefer to go off video now, that's right. Okay. That's fine as well.
Okay. Yeah, thank you very much. I have so much to say, so I hope I will have enough time and yeah, just give you a little bit of an idea what has been done in the 3 Rs and their implementation. So I will start. Oh, why is this not working? So my table of contents, I will start out by talking about the 3 Rs principles and why we should apply them. And then I will give some examples on how the knowledge we are having about the 3 Rs of animal experimentation has been implemented in practice. Then I will briefly touch on some barriers that we still face when moving towards human biology-based animal-free methods, and how we could hopefully overcome them. So why should we apply the 3 Rs? the 3Rs principles are quite old. They were first described by the scientists from Britain, Russell and Birch, and that was 62 years ago now, in their groundbreaking book, The Principles of Humane Experimental Technique. And they had two goals. One was to eradicate what they called inhumanity towards animals by avoiding their use whenever possible and improving their treatment. And also they wanted to improve the quality of scientific and medical research and testing. So the 3Rs principles, replace, reduce, refine, they, when followed, are a tool not only to safeguard animals, but also to improve scientific rigor and reproducibility and ultimately translatability. And it took the principles quite a while to be recognized, let alone to be implemented. Only in the 1990s, they slowly took up or gained some acceptance within the animal research community. But today, they are embedded in many laws around the world. And in Europe, the European Union probably has the most far-reaching directive, because the final goal of that directive, it implements all the 3Rs, but also they want to really move away from live animal use as soon as possible. So I just wanted to briefly say something about the hierarchy. The hierarchy that both Russell and Birch proposed and the EU directive mandates is that replacement should be first priority, followed by reduction and refinement. So that is very important to state here, because to my knowledge in the US, you are not obliged to use a replacement method if there is one, and in the European Union that is different. However, a recent survey actually showed, they surveyed 4 countries in the EU, that the scientists actually found that refinement was way more feasible and pressing than replacement and reduction of animal use. So I will maybe touch on that a little bit later. Then I want to say a little bit about public opinion, because obviously that is a main driver for legislative change. So that's important to see what the public is actually thinking about animal use in science. So I looked at 2 surveys by Pew Research Center, one from 2014 and one from 2018. You can see that the people are divided about the use, but there's a tendency that they don't like animals used for scientific research. And then there's something really interesting that is called Stop Vivisection, and that was a European Citizens Initiative, which is actually a new tool of participatory democracy in the EU. And they were able to collect over 1,150,000 signatures, asking people to support a paradigm shift in the way biomedical and toxicological research has been conducted. So they actually asked for the EU Directive 2010/63, the one I mentioned earlier, to be abrogated and replaced by a way more one focused on really the non-animal approaches, because they were saying they want data that is directly relevant for the human species. And also the directive did not end up being abrogated. The European Commission's Joint Research Center, which is the science hub of the European Union, took action and did a lot to improve the situation. They did a lot in terms of education and training in the 3Rs, also starting already at high school level. And also they have done a lot in collecting and reviewing NAMs, the non-animal methods or new approach methodologies. There's several, what the NAMs stand for biomedical research. And I will show some of those later. Yeah, and this is, that was quite interesting. And this is a recent study from last year, a global study that revealed that the overwhelming majority of people, or almost 94%, think that more needs to be done to replace and reduce the use of animals in testing and research. And I'm just stating this because it's very important to then see how the reality looks in terms of animal use and numbers and so on. Another thing I wanted to first address, I wanted to kind of show why it is really important that we apply the 3Rs principles and really focus on replacing and reducing animal experimentation, because we are in a so-called reproducibility crisis. And yeah, most researchers agree by now that we have that. And also for example, Francis Collins also said that we need to do something to tackle this crisis, because it is a crisis in basic and preclinical research. And if we don't address this, we have a problem, because this is data that then is used for clinical trials, to prepare clinical trials, and that could harm humans. So I just wanted to show some papers that indicate that we are not like other animals. Yeah, for example, for inflammatory disease, it has been shown cancer treatment, the success rate is quite low. Alzheimer's disease is really like basically zero when you think about translation and so on. So that is quite impressive. And also I wanted to point out this book, Rigor Mortis, in case you haven't heard about that book. It came out a couple of years ago, or 3 years ago, maybe 4 now, by the science journalist Richard Harris. And it's really interesting because he lets the research community speak themselves about their problems, and it's really interesting, an interesting read. So why do animal models show poor translatability? We have evidence that many animal experiments are performed are never published. So we have the publication bias that is very large. A large part of what gets published is actually incorrect, they found. It looks like 50 to 90% of the preclinical animal studies are not even reproducible. And in many research fields, we have a translation rate of 0 to 5%, for example, sepsis, stroke, spinal cord injury, traumatic brain injury, cancer, and so on, AIDS, asthma, I could name many more. So we do have a problem. And the other thing is that the National Institutes of Health, the biggest funder and research organization in the world, is actually looking at this problem, and they admit that the selection of the animals that are not well validated models of the human disease under study is a huge issue. Obviously, it's causing costs and yeah, wasting resources, and is potentially dangerous for patients. So can translation from animals to humans be improved is the big question. Most argue in the research community who still use animals that it is due to the poor scientific quality, so mostly due to internal validity issues and poor reporting. Poor reporting is definitely an issue, so they think things can be fixed. On the other hand, there are those insurmountable species differences that also Russell and Birch already described 60 years ago. And yeah, that is still mostly ignored by the animal research community, as a recent survey showed, where they were asked what the problems were, and the species differences were not mentioned at all. So I just wanted to show this paper by Pound and Risquez-Hoitinga that they published in 2018, because it's very important that both internal and external validity have to be good in order to have a translation. And so what is internal validity? That's the quality of the study itself. So that is, you have to make sure that it's well designed, and that is agreed to the scientific standards, and has a low risk of bias. And the external validity is the ability of the findings to be extrapolated or generalized to other research settings, populations, and species. And in our case, obviously, we are thinking mostly about humans. And so the problem, or what the authors conclude, is that they suggest that we can improve the internal validity and part of the external validity, certain settings, but the problem is that the species differences can never be overcome and will always undermine external validity and the reliability of for translation. So I just wanted to have this long introduction because I think it's very important, to lead up to that there are many reasons why we should actually change, and not only one. And as you can see, we are very complex human beings, and we have different habits and lifestyles. And since we consider that about 95 to 98% of human diseases are linked to lifestyle factors, it's really difficult to find an animal that could actually model the situation. So now I come to the 3 R's in practice. Are we doing what we can? Obviously, I can only give you some examples, and I hope that will be interesting. So basically, as Brianne already said, I initiated and co-edited and co-wrote this book, Animal Experimentation, Working Towards a Paradigm Change. I can say later a little bit more what I was thinking when doing it. So what I wrote in the first chapter, I kind of looked at all the different refinement areas: refinement of housing and husbandry, refinement of the culture of care as a potential refinement, experimental refinements, and the refinement of planning, conduct, analysis, and reporting. And this is a video, actually a picture from a video. It's a study by Kathy Shipley from the University of British Columbia. And what she actually did was she trained several rats to be, to do tricks and all kinds of very nice things. And she then exposed animal researchers to those rats, and they could handle them and look at them and get to know them. And the idea was to fostering a culture of care and compassion for those animals. And the result was that they felt a little bit an unease about, if those animals were their research subjects, that they had problems to then kill them at the end of the study because they obviously could become attached. So when animal researchers develop compassion for their research subjects, they do face moral difficulties and moral harms, you could even say, as in the same way the animal caretakers and technicians do. There's this so-called compassion fatigue in this, among caretakers especially, that is obviously from this difficult situation they are working in. But I think that this culture of care is an important starting point in moving towards a culture of compassion for all animals, not just like most people have for companion animals, which then could contribute to their replacement efforts. And then for experimental refinement, I wanted just to show you some studies that were done where they actually assessed the papers and looked for indication of refinements that were applied during the experiments. So experimental refinements is mostly analgesia, anesthesia, humane endpoints, humane killing methods, and so on. Unfortunately, it's very difficult to find, to be sure that what is written in the paper is actually what happened, because we have this problem that not everything is reported. And so for my own research that I did together with Paul Fleckner, we actually decided to take another route. We got access to over 500 animal research proposals from all over Germany, where rats and mice underwent a total of almost 700 recovery surgical procedures. And then we looked at the planned refinements in terms of anesthesia, analgesia, pain assessment and management, health monitoring, humane endpoints, and killing methods. And we had this is a paper where we looked at anesthetic and analgesic regimens. And what was striking there is that for example, the post-operative management was poor. After 30% of those surgical procedures, there was no post-operative analgesia planned. And following 10 of the proposed 10% of the proposed surgeries, they stated that they would give pain relief if needed. but there was not a clear indication that how and they would do, or if they would do a proper pain assessment. So that was not clear. And just to show you some more papers, we also looked at the severity classification of surgical procedures. That's something that the researcher has to do in their proposals. And there it was also interesting because we compared those with, there are catalogs, like pain catalogs, where you can look up what to expect with what kind of surgery. And in almost 60% of the cases, the researchers' severity estimates were lower than these given in the guidance documents that we used to make our assessment. And also the plant health monitoring appeared insufficient to ensure the welfare of the animals in the studies. So also in terms of humane endpoints and killing methods, there was a lot of room for improvement. We found that the killing methods could often be less distressing and less painful. One of the most used methods is still to use carbon dioxide, and that is one of the best researched agents. When you talk about refinement research, they know that it's distressing and there is highly aversive, and still it's used a lot still around the world. It's the most used agent still probably. So yeah, just to give you an idea, these shortcomings have most probably caused avoidable pain, distress, and suffering to these animals. And the general lack of planned use of experimental refinement methods that was identified in this retrospective review of original animal research proposals actually indicates a larger scale problem, most probably not only a German problem. And I could see, things like that in the literature as well, but we weren't sure if they were just underreported or just not happening. And and we thought maybe there's a poor understanding of the significance of refinement to both the animal well-being and the rigor of scientific data collected. So that's another big problem. So what I added to refinement, it's not a classical refinement, obviously, but I just want to mention it because it's so important, is the refinement of design, conduct, analysis, and reporting, because weaknesses in those are really misleading, like they really cause damage. As I said earlier, since legally required animal data forms the basis of decisions to move forward to human clinical trials, the flawed animal research is additionally problematic. So it's very important to educate and train researchers in experimental design, statistical methods, and model selection. It's also important to have a biostatistician that you can consult with. Transparency is very important the whole way, because you should, it's very crucial. What I suggest, and that's suggested by many now as prospective, is a pre-registration of animal studies as it is done for human clinical trials, with their hypothesis and endpoints. And there are two those platforms now available, and one is preclinicaltrials.eu. I advise you to check this out. And also, of course, the use of preparation and reporting guidelines is also super important, as many more things that I won't have time to go into. But then, another thing that is definitely important that I do have to mention is that you should report all study outcomes to avoid traditional reporting bias and selective outcome and analysis reporting bias. So that's really key. And you should ideally retrospectively assess the animal studies. So reduction in practice, I'm not going to talk too much about statistics. Obviously, you have to be really good at statistics, or you have to have a biostatistician that you can consult with, because the idea obviously is not to, when you talk about numbers per group and per study, obviously you have to have that, it has to be statistically sound. You cannot underpower your studies either. But what I'm going to look at here is more the numbers of animals used worldwide. And I will maybe say a little bit more about this, because I don't know who is listening. because in the US, most animals that are actually used are not appearing in the statistic. So what you may know is that the mice, rats, birds, fishes, reptiles, and amphibians are not reported in the US. And so that's what the authors did here, Taylor and Alvarez, is that they calculated the numbers. So it's an estimate. And what they also saw, which was quite shocking, because they did that in 2005 as well. And they saw that there was a 37% increase on the equivalent estimated figures for 2005. So you can see, instead of, if the 3Rs would be taken up better or more broadly, we should actually expect that the numbers would go down, but that's actually not the case. And then also I think it's very intransparent that in the US so many species are excluded, and they are actually thinking that when you compare that to the European Union, when you exclude those species make up for 93% of the animals used. So you can imagine there are estimates that in the US on like per year, it's actually more than shown in this picture. It's actually more like 25 million vertebrate animals are used per year. So another thing, what I wanted to put under reduction or not happening reduction, is that alternatives are not always used, which is a huge problem. As you can see from this graph, because that's an image that came out of a publication from last year, the report from the Commission to the European Parliament, and it shows the animal numbers used between 2015 and 17. And there is progress, but it's very slow, and it's satisfying to see that areas like pyrogenicity testing are finally making major steps towards replacement. While about 170,000 rabbits were used here annually in the previous decade, numbers are down and continuously falling. However, if we think that the methods replacing them have been around for more than 20 years, it's difficult to understand why it took so long to get those numbers down. And there are other examples. At the same time, there are many other examples. I won't go into them now, but just to give you an example, that there are replacements for certain tests, and then you can see in the statistic that those tests are still done, and that should actually not be the case. And the other important thing I wanted to point out here is that the most animals are still used in basic research, 45%. So that is actually an area of animal use that is not required by law. So this is something that a researcher decides themselves if they want to use animals for their basic research study or not. So this is definitely another thing we should think of more. And then I wanted to show some replacements in practice, because I think despite the rising numbers of animals used in science, there's also there are very exciting things happening in the replacement, or I would not even talk about replacement. We should more think about non-animal human biology-based methods, because one important thing to know is that you don't need to replace every animal test with a non-animal test. It's just a different approach to your research question. And so that's, I think, an important thing to say. And that's why most of the times I don't even talk about replacements. I talk about NAMs. So there is a wide variety of NAMs. And as you heard in the last lecture with Dr. Ingber, you got a good idea about the microphysiological systems already. That's very exciting, obviously. Yeah, and since we have the human-induced pluripotent stem cells, that has been amazing 3D cell models. We can also use patient-derived material. That is amazing. So just to give you some examples here, the organs on chips, and they are available also in high throughput format for drug testing, which is very exciting. And then we also have the omics and integrated computer modeling and systems biology and pharmacology. And then another very exciting thing is obviously the AOP framework to gather mechanistic understanding about human diseases. There I just show some papers from Biomed 21 collaboration. That's a very interesting collaboration. Maybe you can have a look at that. And then just a paper that came out a few weeks ago. Actually, the Joint Research Center, they made a meeting or a work group where they used the AOP framework to understand COVID-19. So that's very exciting, I think. And integration of NAMs is key. So it's not possible that you use one of those new approach technologies. You have to really put them together. That's really important. So that's just another thing. Together with colleagues, I wrote this paper in 2019 when Russell and Birch's book had its 60th birthday. And we called it Beyond the 3 Rs. And we said that we have to expand the use of human relevant replacement methods in biomedical research, because as I said to you, this is where most of the animals are used. Basic and applied research together make up for 65% of animal use in science. So that's also what I wanted to highlight a little bit. This is something a graph we had in our paper, just to show that what comes together and how it looks. And then I just wanted to point out that there are many NAMs now to study Alzheimer's disease. Alzheimer's disease, as I mentioned earlier, the animal models had it's going to 0 of translational support success. And then to study breast cancer, there's a lot out there now, also for prostate cancer. Lung diseases, obviously, that's now very interesting. Don Ingwer talked a lot about this, I think, to use the chips also for the COVID research now. And then, of course, the NAMs also to study, the virus, the COVID-19 pandemic. And then, of course, we are still working on technical challenges. I wouldn't say that, just because animals have a poor translation, even if quality of how they are conducted and reported is high. Of course, we have to be aware that we have to, have a high quality in the new models as well. And that's something I wanted to point out because it's a great resource, and I will send Brian some links to the Joint Research Center because they do amazing work actually. So what they did, they reviewed all those disease areas and looked for non-animal approaches, non-animal methods, and they put all those out for free. And there's actually now a database that's actually maintained by a German NGO that is getting all this data, and you can just use the database. That's even easier just than going on the GIRC website. So towards the end, I just wanted to say something about, I gave you a lot of reasons why it's obviously good to move away from animal use. But what is holding us back? That's kind of a big question. And I'm just giving some thoughts on that. And I just wanted to show this again, getting personal with the reproducibility crisis. There I said in the interviews, none of the over 100 scientists that were interviewed mentioned that there could be species differences that could hamper translation. And another interesting paper that came out recently in last year was a paper that found that traditional science is the basis of animal model selection and translational and applied research. So that's another big thing we have to think about. And also I chose this image up here to kind of make the point that we have psychological tendencies that is first confirmation bias, then maybe mixed with cognitive dissonance and motivated reasoning. And this combination is kind of a dangerous mix because we generally look what confirms our beliefs, that's the confirmation bias, and only side with what is most comfortable to not experience cognitive dissonance. And we don't scrutinize contrary ideas. And with that we may impede scientific progress. And there is something called institutional bias, and that's something we have to be aware of and address that within our institutions. So just to discuss the last things, ways forward, how can we really, because obviously I gave you some answers that actually there's a lot that we are not doing. When I look at what we know about, for example, refinement methods and how little of the refinement methods are applied, what we know about reduction, what we know about non-animal approaches, there is so much more that can be done. And obviously it's a long way to go, but I think that we have the society that actually demands that. And of course we have also scientific reasons to move away from many animal models we are currently still using. So the way forward, just some points. Political engagement obviously is important. It's important that the governments actually help here. In the Netherlands, for example, the government was very proactive and said, okay, we are going to set a timeline, and by then, by 2035, they wanted to at least, oh no, actually by 2025, they had a very ambitious timeline. They wanted already to eradicate several animal tests. They had a list for those. But I think this is the way forward. If the government doesn't give you a timeline and action points, I don't think the change is coming fast enough. Legislative change, of course, as you saw, the regulators are not, they are very conservative, and they are not going to allow methods that they are not 100% sure of. So we have to educate the legislators more on the new approach methodologies. Then of course another big thing is the funding. Currently there's so little funding for non-animal approaches, and most of the funding still goes to animal research. In the US, actually, I tried to figure out how much. It was not clear, but it looks like over 50%. But it's not easily, you cannot easily find that out. But I know from Germany how little funding we get for non-animal methods compared to animal research. Then education and training is a big thing, of course, in the NAMs, and scientific collaboration is important, and just to be open-minded. And with that, I'm almost done. I just wanted to point out there is a summer school that we started last year in the US, and we will have the next one next year again. And we are alternating with the European one that is organized by the Joint Research Center. And that was a huge success. Last year we had it online, we had over 600 participants from almost 20 or even over 20 countries. This is that's the NAAT database I was talking about that you can check out. And for early career researchers, this is a really nice platform. They have a Facebook group, and they also have a lot of information about non-animal approaches and grants you can apply for, and all kinds of things. And I also wanted to point out our newsletter. It's not always super interesting, but sometimes you can find here also information on grants, conferences, and so on. And I just wanted to finish with this citation from one of the chapters of the book. And it says, wherever one stands on the animal use ethical spectrum, hopefully all can agree that the conduct of frightening, painful, and lethal experiments on sentient beings, whether mice, dogs, or monkeys, is unacceptable when the research translates in no significant way to human benefits despite decades of effort. Thank you very much for your attention.
Thank you. Wherever you are, I invite you to please join me in thanking Dr. Herrmann for this wide-ranging look at innovative alternatives in animal research that could more fully fulfill the vision of the 3 Rs in a human biology-based science. It's our aim in this lecture series to initiate conversations that move away from either-or debates and to think about win-win ways forward that take the well-being of researchers and their teams, animals, and also maintaining robust research for future studies. Now I'd like to introduce our moderator, Dr. Aaron Cariati. He's a professor of psychiatry at the UCI School of Medicine. He's also director of the medical ethics program at UCI Health. He serves as chair of the medical ethics committees at UCI Hospital and the California Department of State Hospitals. He has also authored books and articles for professional and lay audiences on bioethics, social science, psychiatry, and religion. Hence, Dr. Cariati is located at an important juncture between science, medicine, and applied ethics, very relevant for our lecture series. Please continue to enter your questions into the Q&A box, and Dr. Cariati will try to field as many as possible. Over to you, Aaron.
Thank you, Professor Donaldson, and thank you again, Professor Herrmann, for that terrific lecture and overview of these complex and important questions. So the first question will be my attempt to synthesize one of the submitted questions, and I'll just remind the audience, if you have concerns about wanting to submit a question anonymously, the panelists and myself are the only ones that can view the submitted questions. So don't let that hold you back from submitting a question if you would like. But the first question has to do with distinctions between one animal or one animal model and another. And this is a question I think that could go to any one of the panelists, commenting on it from a scientific perspective, commenting on it from an ethical perspective, or from the tradition of Jainism. So one audience member asked, what about invertebrates? Can we apply the 3 Rs there? They're often considered in vivo models for replacement. And I'm reminded of a bumper sticker that I saw, in fact, today on my way into work, that showed a picture of a primate, a monkey. And the caption was, "I am not a laboratory tool." And it struck me that they picked, this particular species to try to get that message across, rather than picking a lab rat or a mouse, for example. So, is there a meaningful distinction between different types of sentient creatures? I'm thinking of the extreme refinement and delicacy of the Jain monks and nuns that were mentioned in the introduction, of caring even for insects. Does it make sense to try to replace the use of primates with the use of rodents, the use of rodents with the use of invertebrates? Are these distinctions helpful in advancing the kind of goals put forward in this lecture?
I can answer. So yeah, I don't like the definition, because there are definitions out for replacement saying you use lower species. And I don't think this is the way to go, to be honest. And also it's just because we don't know. Like for example, what we do know, and that's actually, they are protected in the European Union, are the octopuses, for example. So that's another species that is not a vertebrate, obviously, but they have special protection because we know way more about them now, and we know that they are very highly intelligent. They can even feel emotional pain. They suffer emotionally and physically. So I think it's our lack of knowledge about other species that leads to using them. And I think we should just focus on human relevant methods. And so we should, that's human biology based. So I would not think we should use other species, not invertebrates either. And cephalopods and decapods are they have already some regulations at least, and I think it will continue. I think I saw something about grasshoppers being more sensitive and than we ever thought, and other things. So I think it's by the day we learn more about animals, but our regulation is not keeping up with the protection of those animals we learn more about.
Kunal, would you want to say anything here?
No, I think those are great points that were made, and that we are still sort of improving our understanding. I also appreciate the point of, do sort of whatever is minimally necessary to do the experiment that is required to tell you what you need to know, whether that's from the FDA or others. Just from a Jain perspective, Jainism does have sort of a clear hierarchy in terms of different types of animals and creatures, and so even the way that you live your life, your goal is to minimize that. And a lot of that is based on this sort of hierarchy. So, even in the lab, I think that oftentimes this is practiced. If you can achieve something with a smaller animal model, or what may be termed as something that has less senses, for example, that's sort of almost always preferred if you can achieve the result that you need to understand.
Thank you, Aaron.
There are a couple of questions here about the use of genetically modified animals in order to make the animal model more applicable to humans, or to move a lower species perhaps closer to the direction of human applicability. Is this a potential step forward in terms of our use of animals in research, or would that represent introducing another harm to animals through what one of the audience members called a sort of dysgenesia, a harm done to the animal by attempting to modify it in this way?
Should I go first, or I don't know?
Yes, Kathrin, most of the questions are for you. Just if there's something specific that is relevant to Kunal, so feel free to direct most of the questions or for Kathrin.
Yeah, so I don't think it's the right way to go at all. If you're interested, there's actually a whole chapter on this topic in our book. It's written by Dr. Bailey, and he looks at especially the new hype also around CRISPR-Cas9. That's like makes genetic modification much easier than it used to, but it's there are a lot of off-target effects that are not talked about much. And the other problem is obviously, think about the animals. The amount of animals you use to generate a new line is immense, and there are a lot of so-called waste animals that are not used because they don't have the right genotype or whatever, for other reasons. So it's a huge waste. And obviously, think about it, the idea is to create a so-called disease model. So this animal is born to suffer. So I'm completely against genetic modification of animals, and I don't think it's the way forward for human healthcare research.
And in general, the concern about instrumentalizing non-human species only for scientific ends, I think was raised by one of the audience members who submitted a comment about the animal training that they were required to take when they first joined a research lab at a major research university. And they were sort of, this person was sort of struck by the rationale that was given for the importance of the humane treatment of animals. And the first reason provided was that if we don't treat the animals in the lab humanely, this could lead to anomalies or fluctuations that might make our results less sound. Right? So the justification was not primarily in terms of the welfare of the animal itself. That was a secondary reason that was given later. But the reason that was highlighted was, for the instrumental purpose for which this animal is being used, if we cause it too much pain or too much distress, that might somehow alter our data in ways that we wouldn't want. So I wonder if, given that in the current climate of scientific research, it seems unlikely in the near-term future that animal models would be, and research on animals is going to be done away with entirely in biomedical science. And if we're thinking about an incremental, stepwise approach to try to make things better, what sort of philosophical shift do we need in our thinking in order to somehow foreground the welfare of the animal itself? And in a way, I'm thinking in a way that could speak to people across the spectrum, right? From you have at one end people who want to say that animals should be given the same rights as human beings in regards to being the subject of research. That's an important position, and it's certainly a position that has a public voice, but at least right now, it's certainly not probably the majoritarian position. But moving along the spectrum toward perhaps a more mainstream opinion, I think most people would agree that, okay, even if animals should not be accorded the same regard or the same rights as a human being, that doesn't mean that we can do anything and everything we want to them. That doesn't mean that they should be sort of purely instrumentalized in a way that a non-sentient tool in the laboratory could be used. How do we reframe these questions or reframe ethical principles so that issues of animal welfare are more foregrounded and not simply a sort of incidental afterthought, such that we have to make the case for them on the basis of purely data-driven considerations rather than on the basis of the kind of thing or the kind of creature that a mouse or a primate is.
Yeah, so to start with, maybe at the beginning where you mentioned, that the most important is that the data is valid and that you handle the animal better or treat the animal better in general because you want to have no interference, no distorted data. And that is actually an argument that I also use to make sure that animals are treated better. It can be seen as a little bit sad that this is that ethics is not enough, that animal concerns are not enough, but it is a big thing. I unfortunately didn't go into that too much, so I recommend really for anybody interested to really read the chapter 1 in the book, because I go into all the husbandry improvements that have been made and what they found out, how to improve the living conditions. But the problem is that the minimal standards that we have at the moment, even in Europe where they are better than in the US, I mean, if you don't even count mice, you can imagine that they are not considered much for their housing either. But even if the best standards according to the EU law are applied, those animals are unhealthy and abnormal because they cannot do all the things they would do. They cannot have display their normal behavior repertoire. That's impossible in those living conditions. So basically, you have to tell a student that the animal there in the cage is already a sick animal. It's not going to produce good data no matter what. And there are many other things that obviously make it worse. If the handling is not correct, if, for example, mice are tail handled, that's horrible for them. Their stress levels go up. All this data we already have, we already know what to do to improve, but it's not done in most institutions. And actually, I just started teaching a refinement course for the first time this year at my university, and I wanted to attract as many people as possible because our School of Public Health, you know, that's not a mandatory course, unfortunately, even so you would think it should be. So I said, yeah, it's to improve the animal welfare and the scientific rigor. And then I had students and they were amazed about you know, what they learned, because when you look at what's out there in terms of best practice, what is known and what is actually happening in the labs, it's such a big difference. And I don't have an answer, you know. I then have to discuss, okay, maybe you can at least ask if you can use 2 different types of nesting material for mice, because they suffer from cold stress because it's way too cold in the laboratory and they can't build their nests properly because they have no material. You know, there's very simple things like that where you're like, this is very weird that we even have to talk about something like that. But yeah, so I think to talk to the PIs, I think they listen more when you say this is going to distort your data. You don't want that, you know, so you have to do something to improve the animals' lives. But at the end of the day, it's all not going to be good enough, unfortunately.
I would love to make a quick comment there. You know, I think from a Jain perspective, I completely agree. You know, I think that the fundamental Jain principle is that every other living being loves its life just like you and I love our lives. And all of these sort of animal experiments, even whether it's just the caging and the environment, not even to describe the disease model or the intervention, all those things, you're performing harm on somebody that loves its life just like you and I do. And so I completely agree. You know, it'd be optimal if we could avoid, these sorts of experiments altogether. One thing I really loved from your talk was, if you're thinking about just reducing as much as possible, at least right now, or can you improve the conditions as much as possible now? I really love the example you gave of exposing people to the rats or the rodents, sort of they're interacting with them, learning about them. You're starting to think of them as another living being that has sort of its own sort of incentives and drives and loves its life, rather than thinking of it just as a laboratory tool. I think that there's a lot that can be done there just in terms of a person's intent and why they're doing these experiments and understanding that. And I think even, that could go further in terms of just training people on how to design experiments properly. I think that there are probably a lot of animals that are unnecessarily used, unfortunately, just because the experiments aren't designed as well as they could be. So, I think I really love that example of, can we sort of help people to understand that these are other living beings and, start to understand that. And I think that can have some very nice benefits in the near term.
Yeah, I totally agree. That's actually another thing we focus on at our center, is to educate and to just teach students how to find the best research model or the best model for their research project. Because the problem at the moment is, and that's why we wrote this paper on going beyond the 3 R's finally after 62 years, is that the animals are still the default model. People just assume that's the go-to model, but why can't we use organs on chips for basic research? We can use all those NAMs now to do basic research. And I really have to say that universities have a huge role to play. They are the animal users, the major animal users, because they do the basic research. So the basic research is not done by the pharmaceutical companies. And as I said, 45% of basic research is 45% is basic research of all animals used. So that's a huge number.
So there was a question put forward that I think is a bit of a comment on what we've been talking about in the last few minutes. Someone asked, would any of the panelists be able to offer their opinion on whether or not science is a field that allows for morality? And I'll just jump in here and say, I sure hope so. And we have examples from the 20th century and even more recent history of exactly what happens when science assumes that it can operate apart from morality, or entirely ignoring moral or ethical concerns, even if those are concerns that are expressed by minority opinions. And I'll mention in this context, because I think it's relevant, the work of James Thomson, who has won the Nobel Prize for his work on 2 major breakthroughs. The first was Thomson was a developmental biologist who derived the first human embryonic stem cell line in 1998. And of course, this raised ethical concerns and was quite controversial because it involved the creation of human embryos and the destruction of human embryos for the sake of deriving stem cells. And there were, of course, people with strong convictions on both sides of this debate. And probably the minority opinion, certainly among scientists, was that we shouldn't be using embryos in the lab in this way. But Thompson, to his credit, didn't entirely dismiss that concern, even if it was expressed by minority. He thought, especially after the birth of his daughter, he's commented on this in an interview, that there's got to be a better way to get these stem cells that doesn't involve us in research that so many people have objections to. And he later became the first person to derive what are called induced pluripotent stem cells. This was in 2007, stem cells from adult tissue that did not require the creation of human embryos in the lab. And this is, to me, this is an interesting example of where attention to ethical concerns and science are not only compatible, but this was a major advance in the science of stem cell research that may not have happened. There may not have been a push in this direction if people hadn't expressed reservation about other methods of deriving stem cells. And it turns out that most of the applications that we now have of stem cells for therapeutic purposes derive from induced pluripotent stem cells, which are not really ethically controversial. It turns out, just scientifically, they're much easier to control. They have lower likelihood of producing tumors, and when you try to apply them to therapeutic purposes. So it seems to me, and I don't know exactly what this would look like, but it seems to me that the concerns about animal testing that are raised not only by the Jain community, but by others concerned with the humane treatment of animals could push us in innovative directions. And there are, I'll mention, there are ethical issues associated with some of these non-animal methods and models as well. Right? If we create a brain organoid in the lab that's sufficiently sophisticated, is that going to somehow become sentient? So it's not that these other models don't have their own sets of questions that they raise, but just as an initial stab at an answer to that question, it seems to me that not only are innovation and ethics compatible, but ethical concerns can often push innovation in really interesting direction. But Dr. Herrmann, I would love to hear your thoughts on this question as well. And how do we situate concern for advancements in research within the context of ethical concerns?
Yeah, that's a very good question, and that was a great example actually for linking those two. I unfortunately think that most of the times ethics is lacking behind innovation. We do things and then we're like, oh, maybe this has some ethical implications. I don't know, just recently there was a news article about Chinese investigators and American investigators. They actually were able to inject human stem cells into primate embryos, and they were able to grow the chimera embryos for 20 days. And that was a sensation in all the news channels. And my first thought was like, what is going on? Because the problem, I'm maybe a little bit pessimistic about our species, but I think the problem is that that as I said, ethics is always lacking behind, and we seem to be so fascinated by technology that I think we do anything. We saw that with the genome editing that was done in humans, with the CRISPR method, and that is not really tested well yet. Of course, I'm not saying that gene editing doesn't have any place anywhere for certain disease treatments and things like that, yes, but not to, for example, create a human monkey. I don't know. I don't understand the chimera fascination, and I'm really scared that this is actually as soon as it's possible, it's done. You know, and I don't know what to say about, you know, people who, you know, bioethicists, how they can contribute better. It's yeah, I think we need more ethical education starting in schools, you know, not only at the university level. And even there, I have to say, I just started the first at our Bloomberg School of Public Health. There was one in-person course for medical ethics, and now I started one for animal ethics. And I think students are just not exposed to all those ethical issues. They just are thrown into their courses and nobody's discussing anything with them. And they also, I mean, I hope it's getting better, because actually we have a program that is really focusing on critical thinking, developing critical thinking skills, because I think as a student, you have to think about everything. You have to assess everything by yourself, and you have to, that's the most important. You shouldn't believe anything. If I were you, I will send you the list of papers I cited today. You should read them all, you know, just to make sure that I didn't tell you anything wrong. I think this is really important, you know, to verify, and not just to be what we, you know, what I said earlier about the confirmation bias, but be in your little group and just tell you each other how great everything is, and that's the truth and nothing but the truth, and you don't see anything else. That's, I think, a huge problem. So yeah, about the ethics, I just think we need more ethics education. We need more discussion in the public about what is actually done in science, because, you know, think about it, that most of the money comes from taxpayers. You know, that's taxpayers' money that we are using also at universities, and we are doing research that's highly questionable in terms of ethics and science.
Thank you. Actually, Dr. Cariati, sorry, thank you. I have to actually jump in because we're at time. Dr. Cariati, thank you for moderating our questions. I really appreciate it. I want to, as we leave, they're both researchers and student researchers. They face a kind of uphill challenge as they consider what would be the next step. For researchers who are on a track to make a change, might feel as though it would really create a barrier in their career, or it might send them in the wrong direction. For students, I think often there is a power imbalance of how to even raise the question. So to Dr. Herrmann and Dr. Parikh, would you have just a first step? What is something for the first step that a researcher or a student researcher might take as they're wanting to explore this alternative for their own future work?
Yeah, so I could provide some resources also for that, because there are a number of courses you can take. They are freely available. For example, the whole summer school that we did last year is recorded, so you can watch that. There are master programs and there are PhD programs that don't use animals. It is difficult and you have to be patient. I don't think it's easy to find, also when you look for an internship, but there are possibilities. So don't give up. I had a student last year in my humane science course, and he didn't want to do animal experiments, and then he got an offer at a very prestigious lab doing mouse research. And I said to him, I don't think this is the right career track for you, because you told me you don't want to do that. And then he looked more and he found something. So there are some possibilities for funding. There's mentorship, and there are certain labs in every university that use the non-animal methods. What's sometimes maybe confusing for the students is that, you know, some labs use animals and non-animal approaches. So you have to kind of find your way through. But I think you just first have to think, where do you want to go with your life, and what's your goal also with your work life, with your career? Because I think we only have this one life, as we know, at least we know of this one life, and I think I assume that most people who work in science want to improve something for others, and so I think there are definitely ways, and the most you have to have endurance, but it is possible, and I can send you, Brianne, some resources that you can share with everybody. You just have to look more, and then you will find your way. Just be critical about everything.
Thank you. And Kunal, do you have any last words?
Yeah, just very, very quickly. I think it's a great and very practical question. I'll share just from my own experience. When I was doing my PhD, I joined a lab where there's quite a bit of animal work that's being done, and all of it is focused on drug and gene development to, ultimately to improve lives. I made it very clear, hey, this is not something that I'm comfortable doing. And so, we had a conversation. We charted out a path for me to provide value to the lab and do novel things that didn't require me to be involved in animal experimentation. And so I would say, for students who are afraid of having that conversation or sharing something like that, if you don't have a PI that's willing to understand that, then that's probably maybe not the right lab for you to be in, or the right people that you want to work with. So I think it's really important in all the work that you do to lead with your principles and what you feel is ethical and moral, and go from there. So I would hope that people have the courage to stay true to that. And I think that if you're in the right place, you'll find the right people to support you.
Thank you. Dr. Erin Cariotti, thank you so much. Dr. Kunal Parikh, Johns Hopkins, thank you. And of course, Dr. Kathrin Herrmann. I want to thank all of our guests today for joining and contributing to this rich space of ethics and innovation between medicine, between ethics, and between a living planetary community. We will plan to have another lecture series next year addressing some important areas of ethical innovation. Do look for a follow-up email in the next week where we will include a link for this video as well as the resources that Dr. Herrmann has mentioned. Thank you again for your presence and for your eager engagement today. Take care.
Thank you. Bye, everybody.
Bye-bye.